everyone, welcome to the ITLS Academy. Today we are going to discuss the topic that is your general principle of food microbiology and foodborne illness. See, what does this food microbiology means? Okay, we all know that the food are what? Food are the substance that provide a nutrition, that provide a nutrition. Okay, so basically the food microbiology are the study of the microorganism which are related to the food. Study of the microorganisms which are related to the food. Okay, what does their structure of that type of microorganism, what is the pH of that microorganism, what is the temperature required for the growth of microorganism. This all things we are going to study in food microbiology. Okay, and foodborne pathogens are defined as the microorganisms which grow in a food and um, when a consumer eat that particular food product, they will become ill. Okay, so basically it is called what? It is called a foodborne pathogen. So foodborne pathogens are what? Microorganisms which grow into the food. Okay, now these all things that we are going to cover at that session. Now come to the next slide. Types of microbes on food. Okay, we all know that there are three types of microorganisms which grow on food. First one is your bacteria, then mold, then yeast. This bacteria are what? Bacteria are the prokaryotic. Okay, and they are only single-celled animal. Sorry, single-cell microorganism. Single-cell microorganisms. Okay, these molds are what? They are a thread-like fungi, and yeast are usually creamy fungi. Okay, the fungi are categorized into into two category. One is your thread-like structure that is called a mold, and one is your creamy fungi that is called a yeast. Now come to the next one. This is the diagram of the mold, yeast, bacteria. See, this is a diagram which we taken with the help of microorganism. What we do, we put a slide into a microorganism and see the different structure of the we set the slide into a microscope and see the different structure of the mold, yeast and bacteria. Here you see that it is a thread-like structure, and we all know that thread-like structure of what they are. Mold. This is your bacteria. Bacteria are different structure. They are either cocci shape, they are either cylindrical shape, they are either dotted shape, rod shape, anything. And now the next one is yeast. Yeast is what? This is your creamy structure. Okay, so these are what? These are the types of bacteria that grow on a food. Now, other type of microorganism which may also concern like your viruses. Okay, viruses are what? They are a protein packets of genes, protozoa. They are complex cell and eukaryotic, and they do not multiply in the food. Viruses and protozoa are not multiplied in the food, but it reaches to the food through a contamination. Okay, so contamination, what? Either they are contaminated with humans, either they are contaminated with water, or sometimes they are contaminated with the other food. Contaminated with the other food. And they all are come under a cross contamination. Okay, so these are what? These are other microorganisms which contaminate the food. Either they are contaminated by the human being, either they are contaminated with the water, or either they are contaminated with the other food. Now, bacteria cell, the first microorganism and the very common type of microorganism that grow on a food are what? They are a bacteria. Common type of microorganism that grow into a food called what? Called bacteria. Okay, they are what? They contain a genes that program the cell and help it survive. Okay, bacteria are reproduced through a binary fission. Binary fission means that suppose this is your mother cell, this mother cells are divided into a two daughter cell. Okay, with and the reproduction are completed with the help of binary fission. Okay, one cell is around one cell can be replicated in 65,000 cells within 4 hours. Okay, because these bacteria are divided in every 20 minutes. So if one cell are present in a food product, then if it kept into a food, if the food are kept into a 4 hour at a room temperature, then the number of the bacteria will reaches around 65,000. Okay, so that's why we are said that food cannot kept at a room temperature for 2 hours. If it is kept, then it will come under a danger zone. Okay, and food will become spoiled. Now causes illness or diseases, that's why it is called a pathogens. Cause deterioration of food, that's why it is called as spoilage. So basically the bacteria which contaminate the food product, they are two types, either they are pathogen or either they are spoilage. Okay, if they are pathogen, then they will create a disease in a human being, and if they are spoilage, then they will deteriorate the food product. And the last one is some useful bacteria are also present and they will help in a fermentation. This fermentation are what? Fermentation are done when the useful bacteria ferment the food product. Okay. Now come to the next one. Some microorganism which causes a disease. Okay. So the microorganism which causes the disease are called what? Are called a pathogens. Okay, because they are capable to cause a disease in a human being. Few of the known microorganisms are harmful to human being. Many diseases can be transmitted from one person to a person or from animal to human. Very few transmitted via food. Okay, so there are few known microorganisms which are very harmful from for a human being, and many disease which are in one person are transmitted to the other person from and from animals to human, and very few are recorded disease which can be transmitted via food product. Okay, so basically these are what they are called pathogens, microorganisms. Okay, so the majority of the microorganism through which the foodborne illness are called. So they are what? Either they are Salmonella, Campylobacteria, Shigella, Clostridium perfringens, Staphylococcus aureus, Listeria monocytogenes, Clostridium botulinum. They are what? They are the microorganisms, okay, which grow into a food and capable to cause a disease in a human being, and capable to cause a disease in a human being. So they are what? Either they are Salmonella, they are Campylobacteria, Shigella, Clostridium perfringens, Staphylococcus aureus, Listeria monocytogenes, and Clostridium botulinum. Okay, now the next one is your food spoilage and food safety. See, this food spoilage and food safety are not all time are same. Food spoilage are what are done by a microorganism. They destroy the food. And if we talk about the food safety, then it is a very broad term. Either they are connected with the microorganism, either they are connected with the physical contamination, either they will connected with the chemical contamination. Okay, so reducing the spoilage can actually increase the risk of harmful bacterium being present. Both preservation and safety must be ensured by a processor. Processor use the hurdle concept to preserve a food and create a safe food. So if we decrease the food chances of the spoilage, then it maybe have a risk that harmful bacteria can be present into a food. Okay, but it cannot cause any spoilage. There are some type of microorganism which cannot spoil the food, but what they do, they release their toxicity into the food, and the food are, when a person see the food from their eyes, they will see that their food are look like a fresh food. Okay, but due to this toxic, the food are spoiled, and when the consumer consume that particular food product, they will become ill. So food preservation and food safety are ensured by the processor, and processor are use the hurdle techniques, in which they are use the more than one type of technique to preserve the food and create the food safe for the consumption of the human being. Now come to the next one. Useful microorganisms are also present. Okay, so these useful microorganisms are used in the food fermentation, like your bread, cheese, wine, beer, sauerkraut, sausage, and other fermented food. Okay, enzymes, antibiotic degrade organic matter. Okay, now bacteria. These bacteria are the most common type of microorganisms which trouble the food processor. Okay, because all type of bacteria, sorry, mostly the bacteria can spoil the food product. Spoil the food product. So this is very concern for a food processor, or they have to eliminate the bacteria from the food product. Now bacteria are what? Bacteria are a single cell living body, so small that individually they can seen only with the aid of powerful microorganism. This bacteria are what? Bacteria are a single cell microorganism, and they are very small, and even they cannot be seen by the naked eye. For the vision of this bacteria, we have to use a powerful microscope so that it can be visible. Okay, now bacteria have a several shape, like your spherical. They are spherical, that called a cocci. Either they are rod shaped, then they are called rods. Okay, now they are, see, this is the first one is your cocci bacteria, this like this. Okay, this is your rod bacteria. So, okay, now reproduction of the bacteria cell. These bacteria can be divided by a binary fission. Binary fission. And in every 20 minutes, the bacteria divided into their two copies. So that's why we cannot keep the food product at a room temperature, that is 37 degrees Celsius for 2 hours, because this is a danger zone for a food product. Danger zone for the food product, and food becomes spoil. Okay. Cell in the active stage of growth and metabolism are referred to as a vegetative cell. Now see, this is a division of the bacteria. This is your bacteria, cocci bacteria. Here the cells are started to division. Here the cell wall are made. Then this divided into a two part. This is your rod shaped bacteria, and here the division. Now the next one is your, under a favorable condition, each cell divide can average about once every 15 or 30 minutes, or approx it is said that it is 20 minutes. So there is a concept of each cell of bacteria can divide into a 20 minutes. So if we put a food at room temperature for 2 hours, then it will, there is a millions of bacteria now present into a food product. Okay, see, this is a zero reproduction of a single cell. bacteria single cell, and the see when we put into a 0 hours, okay, this is your rod shape, this is your foci. Okay, see the number increasing, and in 15 hours it is 1 billion. Okay, if you put into a 3000, it is around more than 4 lakh. Now the growth curve of microorganism. Okay, this is very separate topic that we discuss in a detail. Here what we are going to do, we are see the basic of it. What happened when the bacteria start dividing? Then it first phase is your lag phase, where the growth does not occur. Then the exponential phase are there when the bacteria growth are occur when the time are increases. Then this is a stationary phase, and after that death phase are there. Okay, now the next one is your mold. Mold are what? Mold are a multicellular microorganisms, and they form a tubular filaments or a mycelia, and they are branch. Okay, and they are reproduced through a fruiting body or a spores. Okay, suppose that this is your bacteria, sorry, this is your mold, and here the spores are formed, and they will detach from the mother body and form a new mold. So this is how this spore reproduction occurs. They are larger than the bacteria and longer than the yeast. Okay, they are widely distributed throughout the environment. They are also present in a soil and in the dust. Okay, now will grow on almost any food. Okay, they like your pastes, paper, plastic, paint. Okay, these molds can be grown in either food, and along with the food they can also grow into a paste, paper, plastic, paint, etc. So when we have to cook the food, then we have to take a one more precaution. We have to clean the environment also. Clean the environment also. Okay, and they can tolerate even a cold temperature. Little heat resistant, they required oxygen to grow. Okay, no growth in canned food unless air remaining in container and loss of vacuum. Okay, in we all know that in a canned food vacuum are there. When vacuum are there, then mold cannot grow. Okay. Now the next one is your yeast. Yeast is also what? It is a single cell and microscope living microorganism. They are oval shape, egg shape, and they are smaller than the mold, but they are larger than the bacteria. Okay. So if we talk about the size of the microorganism, then the mold are the bacteria are the smallest microorganism. The bacteria are the smallest microorganism, then the yeast, and after that mold. Okay, they can reproduce by a budding. Okay, see birds are formed. This is your this is your yeast. Here the birds are formed, then they will detach from the mother body and form a yeast. Okay, yeast are also widely distributed into a nature. They are often associated with the liquid food containing sugar and acid. They can tolerate even a cold temperature, and most heat are destroyed by heating at 170 degree Fahrenheit. Okay, now some intrinsic and extensive properties are there which affect the food safety and spoilage. Intrinsic property that will consider as the inside property of the food, and the extensive that will consider as the environment, yani outside of the food. Intrinsic property include pH, moisture, oxidation, natural antimicrobial constitute, biological structure. Extensive properties like your temperature, relatively humidity, presence of gases, antimicrobial or added microorganisms. Okay, now preferred consideration for the bacterial growth. Okay, fat term. See, this is a basic thing that we have to learn this. This is the basic requirement which have a bacteria required for that for their growth. That is your fat term. Fat means food. A for acid range, T for temperature, T for time, O for oxygen range, and M for moisture. Okay, if bacteria have these six factor, then they will grow very fast. Okay, foods include, like, if suppose that if a food contain high amount of carbohydrate, protein, and fat. Okay, if the pH of the food is in between the 4.6 to 9. Okay, temperature are between 5 degrees Celsius to 63 degrees Celsius, they are called a danger zone. Okay, and time minimum 2 hours. Okay, and the oxygen varies by a microorganism. Moisture above 0.85, water activity. Okay, so that's or suppose that these are the factor which this is the variable condition which is required bacteria for their growth. Suppose that we have a chicken piece. We all know that this chicken are what? High in protein. Okay, so bacteria have a first condition for their growth, that is a food, protein, which are present in a chicken. Now we put the food into a range of temperature between 5 degree to 60 degrees Celsius. Okay, now the time is also put a food for 3 hours. See, this requirement are fulfilled, this requirement are fulfilled, this requirement are fulfilled, and even the moisture content in a chicken is around 80%. Okay, this requirement are fulfilled. And when a food put into a outside the refrigerator, then the oxygen requirement are also fulfilled. So bacteria get all the condition and they will spoil this chicken piece very easily. Okay, if we cut any one of these, then the chances of the chances of the bacterial growth also decreases. Okay, so many foods like your milk. Okay, milk, when they are in normal condition like liquid state, then they will spoil 2 to 3 days when they are kept between a 0 to 5 degrees Celsius. Sorry, 0 to 5 0 to 5 degrees Celsius. Okay, but when we remove the moisture from the food product, this milk, okay, at that condition the moisture content will decreases and it will become a powder state. Then we can store this food product at a then we can put a food product at room temperature even for a 6 month. Okay, so these are the conditions that affect the food product, and this is how we can change the condition according to them. Now come to the next one. What microorganism need to grow? Okay, microorganism required water, required sources of energy, required nitrogen, required vitamins, growth factor, and minerals. Okay, now the next one is your oxygen requirement. Okay, the oxygen are required by the for the aerobic microorganisms, and there are some microorganisms which can be grow in the absence of the oxygen, they are anaerobes. But the majority of the bacteria neither restrict the aerobes nor restrict the anaerobes. They are what? They are called the facultative anaerobes. They can grow even in the presence of the oxygen and even in the absence of the oxygen. Okay, so water activity of the some common food like fresh water, they have more than 0.95, liverwurst 0.96, cheese spread 0.95, caviar 0.92, fudge sauce 0.83, semi-moist pet food 0.83, salami 0.82, soy sauce 0.80, peanut butter 15% total moisture, that is 0.70, dry milk 8% total moisture 0.70. Now pH is the next one which is also required for the growth of the microorganism, and this pH are measured in a acidity. pH is equal pH is defined as a minus log of hydrogen ion concentration, major control point for a bacteria. Okay, if we change the pH of the food product, then the chances of the growth of the microorganism will also affected. It can even if greater impact on the quality of the food product. So classification food can be classified on the basis of this pH. Either they are acidic food, low acidic food, or alkaline food. Either what they are acidic food, they are low acidic, and they are alkaline. What they are? They are classification of the food on the basis of pH. Now the next one is your pH. pH ranges for a food. Okay, so 0 pH is for a limes, 2 pH is for a fruits and berries, and pH 3.5 is also for a fruits and berries. pH 4.6 is for a bacterial growth. At this pH, if a food kept at pH 4.6, then this is a best pH for the growth of bacteria. Okay, now pH 5.5 is for a veggies melon, and pH 7 is neutral, and this is for a meat and milk. pH 9 is also for a meat and milk, and pH 10 is for a bleach solution, and pH 14 is for nothing. So this is a pH range for a different food product. Now comparison of the bacteria requirement. Yeast comparison of a growth of requirement of different type of microorganism. Bacteria required pH at the range of 4.4 to 9.8. Yeast required 1 to 11 pH, and mold required 1 to 12 pH. What bacteria required water activity more than 0.85. Yeast required water activity more than 0.60, and mold required water activity more than 0.60. Okay, bacteria sometimes required oxygen and sometimes don't require oxygen. Yeast don't require oxygen for their growth, and mold required oxygen for their growth. Temperature of growth classification. Okay, according to them, there are psychrotropic, mesophile, thermophile, and extreme thermophile. Okay, the temperature at a lower temperature when a microorganism are grow, then they are called psychotroph. Okay, then the temperature when a temperature are between a normal temperature and at that temperature microorganism can grow, then that is called a mesophile. And when a microorganism can grow at a high temperature, then they are called a thermophile, and extremely thermophile when they are grow at very high temperature. Okay, so if we see the optimum temperature for a psychotroph, then they are at the range between 14 to 20 degrees Celsius. Mesophiles are what 30 to 37 degrees Celsius, and this 37 degrees Celsius is even at a room temperature. Thermophiles required 50 degrees Celsius to 66 degrees Celsius. This is a classification of bacteria by a temperature range for their growth. Now spore forming bacteria and non-spore forming bacteria. Most of the bacteria exist only at their vegetative cell. Okay, all cocci and some rod shape. They are what? They are a non-spore formers. Some group of rod-shaped bacteria produce spores, and they are called as spore formers. Exist as exist at both form. Either they are vegetative cell, they are spores. These are the diagram of this. Okay, now bacteria spores are dormant stage in normal cell growth and can be germinate and produce new vegetative cell. Ability to survive a wide range of unfavorable conditions, spores in mold and yeast, and generally they are not resistant. 3 microscope form of spore forming. Vegetative cell, vegetative with spore, spore. Okay. Now, resistance of spores to environment. In general, bacterial spores are extremely resistant to heat, cold, chemical agent, and they can survive in water boiling if they put in less than 16 hours. Every microorganisms have required a optimum temperature for their growth. Microorganism group according to their relationship to the temperature. Okay, psychotrophs are what? Psychotrophs can be grow even at a low temperature. Here the psychotrophs are come from two word. One is your psycho, which indicate cold, and trophs growing. Okay, and the optimum temperature is 58 to 68 Fahrenheit, that is your 60 to 20 degree Celsius, and grow slowly on food at a refrigerated temperature, that is 40 degree Fahrenheit. And primary they are come under a spoilage microorganisms, and few of them are pathogenic. Mesophilic can be grow at a normal temperature. Mesophils are also made up of two type of word. One is meso and another one is your phile. Meso means middle and phile means love. And the optimum temperature for a mesophilic microorganism, mesophilic bacteria are in the range of between 86 degree Fahrenheit to 98 degrees Celsius, that is 32 degrees Celsius to 37 degrees Celsius. All microorganisms that affect the food safety can grow in this temperature range. Clostridium botulinum is a mesophile. Then the thermophiles, also made up of two type of word. One is your thermo, that is for heat, and phile for love. Many or spore forming bacteria obligate thermophiles. Spore will not germinate and grow below a 122 degree Fahrenheit. Facultative thermophiles and growth occur at 122 degree Fahrenheit to 150 degree Fahrenheit. Some of these bacteria are heat resistant and they can survive more than 60 minutes even at a temperature around 250 Fahrenheit. Thermophil bacteria are not pathogenic and they do not affect the food safety. So these are the basic or general of the food microbiology and the foodborne pathogen. So, thank you for watching. Please like, share, and subscribe. If you have any query, just let us know in our comment box. You can also ask your query to the helpline number that I already mentioned in your account. Follow the ITLS Academy on different platforms: Instagram, Facebook, Twitter, YouTube, WhatsApp, LinkedIn, and you can subscribe our channel ITLS Academy on YouTube and get our app from a Google Play. Thank you so much. If you have any query, just let us know in a comment box.